Okay, here we go with part two of chapter 14. Um, where we left off was now have a functional piece of RNA that is supposed to code for the protein that we're trying to build. But before we can actually go and turn this into a protein, in all organisms that are eukaryotic, we actually go through another process. And that process is this RNA processing. So we take the RNA and we do a few things to it. So the first thing we do is we add things on either end. And this is simply just a way to make sure that the RNA molecule lasts a little bit longer. Now, RNA molecules are not supposed to last forever. They are temporary because you want to build the protein for a little while and then stop building it. But in order for it to last long enough that it will make a difference and make enough proteins, you put these little caps on the end, and those caps are a little protection because that's where the molecule usually starts to deteriorate from. So they're different. The 5' end gets a 5' cap, and the 3' end gets a whole bunch of A's that are put on there. And then the really interesting part is this RNA splicing, this idea that you actually take a molecule that is usually much longer and you cut it down. What is happening here is you have these series of in the RNA molecule, you have these sections that are called exons. They're the things that you want to keep, and then there's these introns that you want to remove. The best way I usually tell people to remember is the introns intrude into the code, so you got to get rid of them. So we got to get introns out of there, and so we use these things, and this is one of the funnier names in AP Bio is SNRPs. SNRPs are these very strange molecules. They're a mixture of RNA and proteins, and they actually will cut the RNA down to size, and they will take out a section in the middle and then they'll glue it together. So they use this is called splicing. So in the old days when you actually made movies with film, in order to make two scenes fit together, you'd film one and then film the other, and then you take literally the film and you would glue them together. That was called splicing. So that's essentially what's going on, is we're cutting out the middle and then we're gluing two ends together. The interesting thing about SNRPs is that they're ribozymes, these weird things that are both RNA and enzymes. And it's one of those, we've talked about this idea that in the early life, there may not have been DNA. It was simply a RNA world. So, but those SNRPs are very important. So here we have that full big molecule, and what we're going to do is cut out these little pink regions and cut it down to size. So that is the idea of this. This is showing you that spliceosome, those SNRPs and the other proteins that actually come in and they cut it and then they glue it back together. Okay, and this type of arrangement was never really well understood. The question of what is going on here, why is this even important, is really still a mystery. The leading ideas behind this are that one, it is a way to actually alternatively use the same gene to make a slightly different version of a protein. So a good example of this is antibodies. Antibodies in your body are not coded for with completely separate genes. What you have is one gene, and if you cut it one way, you get one version of the antibody, or if you cut it a different way, you get a different version of the antibody. So that's the idea of alternate RNA splicing. And the other idea, it's a really complex idea, but really neat idea, is that what this is doing is actually the protein may have different regions to it, and those regions may do different things. One region of the protein may attach it to the cell membrane. Another may be the active site. Another may be an inhibitor site. And what it allows is those separate areas of the protein to be physically connected on the genes, but separated so that if you change one, it doesn't change the other domains. And this would possibly allow different parts of a protein to evolve separately. That's a really difficult concept to get and difficult to explain, but there's your protein, and this is like sort of the separate regions, and they're coded for in different areas. And so if you had a mutation in this middle one, it's not going to affect this other part. So we could change this area of the protein without affecting that area of the protein. That's the idea behind that. All right, translation. Translation you have played with before. It involves a couple of things. tRNA is one of them. tRNA is transfer RNA. It's not translation RNA, but it's a small RNA piece that has two important parts. It has an amino acid attachment site. There's my dog. Sorry. And an anticodon. Anticodons match up with codons. So you don't actually need 64 tRNA to match with the 64 codons. One, stop codons don't have tRNA. But also, where we have those overlaps, where you have that last letter, you can actually have an anticodon that has a wobble letter for the last that will match up with anything. You also need this molecule called aminoacyl tRNA synthetase. That's the thing that actually loads all of these tRNA with the correct amino acid. So you have to have 20 different ones, one for each amino acid. And then the ribosomes. Ribosomes consist of two subunits. Finally, something easier. They call it the large ribosomal subunit and the small ribosomal subunit. And then you have these sites along there that allow this to work. And so I'm going to try to zip through. This is a tRNA. This is one version of it. And the only real important part is this part up here that attaches to an amino acid, and this part down here called the anticodon. This is a different way of looking at it, three-dimensional. This is a cartoon version, but again, the two most important parts, anticodon, amino acid attachment site. And everything's going to match up the right way. So this is showing you how the tRNA is loaded. We take the tRNA, we take some ATP, we put it together with the right amino acid, and now we have a loaded tRNA that's ready to go, or charged is another way to put it. And then this is the ribosome. Looks very complicated, but basically it has a couple of important features. One is this is the RNA. There's a little channel that the RNA can sort of run through the ribosome. And then there's these slots called EP and A. It doesn't stand for Environmental Protection Agency. It's just some weird letters. The only one I actually remember is the E stands for the exit site. And then there's this spot, this little tunnel that allows the growing amino acid chain, the growing protein, to actually exit there. This sort of shows you the idea, and actually I'm going to skip right ahead to this. So this is how it first happens, that we load this messenger RNA with the first one, and the first one is always the same. You got the start codon. Matches with the AUG is the start codon, the anticodon is UAC, and it's always methionine. So it's every single protein starts that way. We can cut that out later. But then we load it up and we make the full ribosomal subunit there. And then what happens is we slowly start, it actually happens very quickly, but we start loading this A slot with the next amino acid. So this anticodon right here will match with that codon, and then this is carrying the right amino acid. So brings that in. So we load that, and then what happens is the amino acid chain gets transferred over. And so what we essentially have done is grow the amino acid chain by one amino acid. Okay, and then everything slides over. So this tRNA goes to the exit slot, and of course exits. That opens up this A slot, and that allows this whole cycle to start again, where the next amino acid can come in. And every time you go around this circle, you grow the amino acid chain by one. It seems like it would take forever this way, but this happens extremely quickly. This entire process takes about a third of a second. So within a minute, you could make an amino acid chain that is 180 amino acid chains, 180 amino acids long. So the size of most proteins. When you get to the end, there's no stop tRNA. There's nothing that matches that, so you get this thing called release factor. It comes in and sort of blows the whole thing up and frees the polypeptide to go on. So one thing you can do to make it more efficient to make protein is you can actually have multiple ribosomes working on the same RNA. RNA molecules are long, so as soon as the ribosome sort of clears the beginning part, you can add another ribosome. And so there's a couple things here. This is an example of the multiple ribosomes working off of one one messenger RNA, and there's the actual image of it. Okay, signal polypeptides. So a lot of times the first couple amino acids are actually not meant to be part of the final protein, but they actually signal where it's going to go, and that's this idea of signal recognition particles. But I'm not going to get into that because it's a little complicated. Mutations. You guys had a nice little POGL on this, and I think it makes that should be pretty self-explanatory. But base pair substitutions are a really common one, and basically you're just replacing one letter with the other. Sometimes that can have no effect, especially if it's the third of one of the codons. So the third letter of the codon usually would not necessarily affect it. Sometimes it can do something called missense, which means you just replace one amino acid in the protein. And there's two types there. There's what's called a conservative and non-conservative. Conservative means you're replacing it with a similar amino acid, and so you might not affect the protein that much. Non-conservative, you would replace it with a different type of amino acid, like you go from a polar to a non-polar amino acid. That might make a problem. And then nonsense mutations are where you actually end the production, so you put a stop codon in there, and that obviously messes it up, and you don't make a useful protein. Insertions and deletions are much more problematic because not only do they change the one amino acid there, but they change everything after it because everything is moved over. If you insert one letter, that three letters gets shifted over and everything gets messed up. And so it causes a really big problems unless the insertion or deletion is in a multiple of three. So if you insert three letters, then you're just going to make a protein that's one more amino acid long, and it still conserves that reading frame. Anything that causes mutations is a mutagen, and a lot of times some of our cancer-causing things are caused by mutagens or the inability to proofread our DNA, so causing more mutations. So here's some examples, but I think this is well covered in the POGL, so I'm not going to go through that. And this is a nice summary of everything here, showing you everything from transcription to RNA processing to the translation here. So this is a good one to look over and check out. And that is the end. So thanks for paying attention, and we're going to do a few more activities. I know this is confusing, but I'm going to have some activities on Friday that are going to help out with this one. Thanks. Bye.